Okay, welcome to biological physics, physics 3250. And I just started the video. We're going to be video recording every class. So if you miss a class for whatever reason or you want to review something, you can watch it on Moodle. This means the video camera is pointing up this way, so you guys won't be seen on the video. But if you probably don't want to raise your hand and say, my name is such and such, and then start a profanity-laden stream, because it will all be recorded. But other than that, I think everything's fine. Okay, every class I will put over in this corner of the chalkboard assignments for the future days, just to let you know what's going on. So we'll start right in on chapter 1. You'll have a homework not due this Friday, but a week from Friday. So we'll be talking about that as we go. All right, so I think the first thing we want to do is find out who everybody is. So I will we'll go through the list I have here, and I want you, when I call your name, I want you to tell me a little bit maybe about yourself, like in particular why you're taking this class. So is Amber Anderson here? No. How about Samad Azuz? Now also tell me if I've mispronounced your name.
It's fine.
Okay.
It's needed for my major.
Needed for a major. What is your major?
Bioengineering.
Bioengineering. How many of you are bioengineering majors? Yeah, I thought the majority of you would be. Any physics majors? Okay, couple. Anything else?
Mechanical engineering, biochemistry.
Biochemistry. Okay, well, I think this would be a good class for everybody, but I think the majority of you, it seems to me, were in the bioengineering program, and that's great. I think this would be a good class for bioengineering. Okay, Rebecca Barbera. Did I pronounce that right? Okay. So what year are you here at Oakland?
I think technically senior. It's like my 5th year, but only my 4th year at Oakland.
Okay.
I did home first.
So how many of you are in your last year at Oakland? Last year. How about your 3rd year, say? 2nd? Anybody here their 1st year?
I believe I'm going for a 5th year.
Okay. All right. Well, we have a mix. That's good. Okay, Olivia Bolton. Rita Butchlaz. I'm sure I mispronounced that. Carrie Davatilla. Samantha Garcia. Okay, Samantha. Do you have the textbook yet?
No, that's
Okay. Yeah, I wanted to mention that the textbook, I said it already in the email. Here's the book. If you want to use PDF versions, you can get it from the OU library. It won't cost you a penny. You can get it through the OU library, a soft cover copy for $25, or you can go to the bookstore and you can pay full price. Try to get the 5th edition. The homework numbered problems are numbered differently in different editions. So if you do get an older edition, you better get the PDFs just to do your homework. Otherwise, you'll do all the wrong problems. There's a website for the book. Let me just write down what that website is. It is HTTPS. This has changed recently.
Is the website also available on Moodle?
Probably. I hope so. If it's not, I'll put it up there. This is view. That's the book's website. And on that website, you can get several things, such as the errata. You know, we work and work and work to try to make sure there's no errors in the book, and then it gets published and we find a whole bunch. So you can find the errata. I would suggest you download it and mark in your book. I don't usually like to write in my book, but if you have a paper copy, make the corrections so you don't get stuck. There's also, this book has something unusual. It has a blog that goes with the book. That is, let's see here, the hobby raw blogspot.com. It's updated every Friday morning. So it's not required reading, but it's just extra auxiliary material for the book. So any questions about the book? Okay. Then let's continue on here. And where was I? I just did Samantha. So now I'm doing Yosef Gara Koza. Okay. Greta Gashai. Close enough? Okay. And Greta, you use Moodle?
Yes.
Okay, everybody use Moodle? All right, I have Moodle for the class. Do not use the email that's within Moodle to try to get a hold of me, because I don't check that Moodle email much. Just use my standard OU email, raw@oakland.edu, and I should be able to get back to you pretty quickly that way. So you're welcome to try to get a hold of me anytime, but email is probably the best way to get a hold of me. Okay, so now Connor Halloran. Okay, Connor, do you like to go to office hours?
Yeah, when I get a chance to.
Okay, good. I think that students should go to office hours more. My office hours are right after this class, but if you come by anytime I'm there, I will stop and talk to you unless I have some meeting I absolutely have to go to or some deadline that's just I can't get out of. I'm a morning person, teaching 8 a.m. class, but if you come looking for me like 4 or 5 in the afternoon, you may not find me. But you can find me even before class if you come down to my office, 166 Hannah Hall. So I encourage you all to come. You're certainly welcome to come. Okay, Brett Heglin. Brett, do you like to do your homework in groups?
Haven't really been a group person.
Okay. Well, the good news is you can do it either way. You can do it in a group, you can do it by yourself. But what I want to mention is there's no problem with working together on the homework. Now, I have a problem with you just taking somebody else's homework and just copying it. But if you're working with somebody, even if they're a little ahead of you, so you're getting more out of the collaboration than the other person, that's okay. I don't have any problems with that. You can work on your homework together. It's not against the rules. Hand in your own assignment. Don't hand in joint assignments between various people. Okay. Cameron Johns. Mark Kamash.
Right.
Mark. Okay. Do you like to read the textbook before class?
For me, it's better as like a second exposure rather than a first.
Okay. You can do it that way. I would encourage you all to read the text before you come. You gotta read the text at some point for sure. But I will encourage you by putting up the reading assignments, trying to be as specific as possible of what sections we'll be covering for the next class. So if that works for you, try to read before class. If it works for you better to read it afterwards, that's okay too. I have in other classes done little quizzes at the start of class to make sure people read. We won't be doing anything like that, so don't worry. So where are we now? We have Ivana Marceta?
It's Ivana.
Ivana. Okay. Ivana. Let me just write that down. And your question is, do you like to have surprises when you take an exam? I didn't think you would. I don't think anybody does. And to prevent that, what I'll be doing with our exams is I'll be giving you practice exams before the exam. And before the exam, we'll cover and go over these practice exams in class together. So hopefully that will reduce the number of surprises. Now, sometimes students still think, you still get surprised, and sometimes it's hard for me to predict what will surprise the students. But I'm hoping that these practice exams will make it a lot simpler for you to prepare for the exams. Alexandria Martin. Trevor Olson. Trevor, do you like to have a comprehensive final at the end of the class?
Do I like to?
Yeah.
Not in all cases. No.
Not in all cases. All right. Well, good news and bad news on this. You will have a comprehensive final. That's the bad news, probably, for most of you. The good news is that, first of all, it's not worth a very large percentage of your grade. And second of all, the comprehensive final will just cover questions from the old exams and practice exams, verbatim, not even change numbers. And you'll get solutions to the exams and practice exams. So that comprehensive part really rewards just hard work. You'll know exactly what's going to be on the test. But like I said, that's only a small part of the overall class grade. And also it is, you have several practice exams, so it adds up to quite a few problems by the time you add them all up together. Okay, where are we? Augustin Perez Chavez? Okay. Now, have you ever taken an introductory physics class?
Yeah.
Okay, good, good. I'm assuming all of you have taken introductory physics. If you haven't, you probably ought to talk to me, because I'm not going to develop everything from first principles. We will. This class is going to be applying what you learned in introductory physics to biology and medicine. At a somewhat higher level, probably, but not a lot higher. So I'm expecting you've had introductory physics. If it would, all the better if it was calculus-based, but if it wasn't, it's not going to be a big disaster. Jonah Ragel. Okay, Nicholas Schneck. Okay. Have you ever taken calculus? Okay, good. Calculus is going to be also required. We're going to be using calculus quite a bit. So I would expect you've had a year of calculus in this class. That being said, the calculus that we actually do, it's going to seem at first like it's high level, because sometimes we'll be talking about partial differential equations. But all we really are going to be doing is we're going to develop the differential equation, and we're going to stick in a guess solution, and we're going to take some derivatives and maybe find some constant coefficients. It's not going to be a real high level. Certainly no theorems, proofs, or anything real abstract. So if you haven't had calculus, you might want to come talk to me. We'll try to figure out if this is the right class for you. But if you had a couple semesters, you know how to differentiate and integrate, you should be in okay shape. Alrighty. Isaac Philander. Did I say that right?
Yeah.
Okay. Have you ever taken biology?
Yeah.
Okay. Surprisingly, introductory biology is probably less important than introductory physics and introductory calculus, because I'm a physicist and I tend to stress the physics. But if you've had biology, so much the better. Like I said, you'll get by, okay. You'll learn a little bit of biology, but all the better if you've had introductory biology. Okay, Lily Turr. Lily. Let's see, which are you more interested in, biology or medicine? Biology. Okay, how many of you are main interest is in biology? How many in medicine? Okay, well, let me just do a quick advertisement. We do have a second semester of this class, which is medical physics. The biological physics that you're in is more looking at how physics applies to physiology and to biology in general. And then the second semester is going to be more medical imaging and different ways physics can be used in medicine. So you don't have to take one to take the other. But I just wanted to let you know, I won't be teaching the medical physics part next semester, but I would encourage anybody to take it if you enjoy this class. All right. And Ethan Barastrate. Okay. Well, I don't really have a good question for you here, but why did you take the class?
I needed a science elective for mechanical engineering.
Okay.
And then I like physics and I like physiology, so it's cool to learn how like the body works with physics.
All right. You'll probably like the very beginning of this class most. On Friday, we will be talking about muscles and biomechanics. So, but yeah, hopefully you'll like the whole thing. But anyways, we'll start right in with some material that'll interest you. Now, some of you came in later. Some of you weren't on the list. Is there anybody who I didn't call on who came in? Yes.
My name's Carrie.
You need what? Okay. Carrie Davatilla. How do you pronounce it?
Davatilla.
Davatilla. Okay, that's easy enough. Very good. And yes.
Olivia Bolton.
Olivia Bolton. Okay. Very good. Yes.
Amber Anderson.
Amber Anderson.
And Yusuf Kanakoza.
Say it again.
Yusuf.
Yusuf. I remember there. Yusuf Yaria Quota. Okay. Anybody else? All right. Well, very good. Now let's talk a little bit about the syllabus. Did everybody get a copy of the syllabus? It's right here at the end of it. Let's see. Anybody need it? There. Pass those. I think that'll be enough. Oh, we'll need one more. All right. A lot of this we've already talked about during the introductions. But, again, my name's Brad Roth, and my office is 166 Hannah Hall. You got my email address there. We have this office hours. And yes, this was on the syllabus. Okay, we talked about the textbook. The grades. We're going to have mainly grades on the exams. I actually changed things a little bit. I've been teaching this course here at OU so much that I've de-emphasized the homework a little bit because I'm assuming that you guys probably have friends who have all the homework solutions. So homework's only going to be worth 5% of the grade now. And so a little higher previous semesters. And also the comprehensive part of the final exam, which is questions from all tests and practice tests, is going to be worth 5%. And then you'll have 3 exams, one of which will be on the final exam day, but 3 exams covering different chapters, each worth 30% of your grade. So 3 exams, each at 30%, plus a 5% for the comprehensive part of the final, plus 5% for the homework. That's 100%. And you have the schedule on here, and you have something that's a very detailed schedule on the very back sheet. I'm going to try to go mostly paperless this semester, so this is hopefully the only paper you get from me. This detailed schedule looks a lot better in the color version, so you might want to download the PDF because then it's all color-coded, what's homework, what's reading assignments, and so on. And you do have all the homework assignments written on here, so anybody who feels ambitious can do all their homework this weekend and be done with it. Not that I would recommend that, but you know when they're all due, you got the dates of the exams, exactly what sections to be reading for the exams. So, questions about the syllabus? Yes?
So how is the homework based? How is the homework graded? Is it graded based on completion, accuracy, or both?
Well, I grade it for accuracy. None of them are multiple choice or anything like that. They're all problems to solve. So I will give partial credit if you're on the right track. You make a little screw up right at the end, I'll give you 9 out of 10 points. If you kind of lost halfway through, you may only get half credit. If you don't do anything, you get 0. But it's all problem based. The homework hopefully will reflect similar to what the exams will look like. And you'll get the solutions afterwards. Does that help? But definitely, I will be grading the homework. I won't just give you a check mark if you hand it in. Yes.
What's the grading scale look like for letter grades?
I generally grade on a curve. I'll guarantee you this: 90, 80, 70, 60 for A, B, C, D will be the most stringent it will possibly be. Okay, so if you get 90% average, you get an A, some type of A. But usually, I like to give kind of a more difficult test than a big curve, so there's a good chance that it'll be more generous. I think every semester I've taught the class, it turns out to be a somewhat more generous curve than that. And I'll tell you what sort of the curve is for each exam so you'll kind of know where you're standing. But yeah, if you get it 98, 70, 60, you'll get guaranteed A, B, C, D. Other questions? All right, when my dad was in college, he went to a math class, and the first day of class, the instructor said, ask questions. He said, you got to ask a lot of questions. So he went in the second day of class, and the instructor said, looked out in the class and said, any questions? Everybody kind of stood there, sat there like you guys are. And then the instructor said, class dismissed. And they learned really quickly that they had to ask questions. So I'm not going that far, but I want you guys to ask questions throughout, now or throughout the class. You should feel free to chime in. Okay? Now, we're going to go ahead and start on the very first part of the book, which is in chapter 1. The first section is about distances and sizes, and we're going to do a little discussion of that. And this kind of material can be on the exams, as you'll see when you get the practice exam. I may ask you for the size of something. How big is a red blood cell? How big is a E. coli bacterium? And you'd have to answer it, but only within a factor of 3, bigger or smaller. So what I'm trying to say is, I want you to know order of magnitude how big stuff is. I don't care if you know exactly how big something is. But if you don't have a kind of an order of magnitude idea of the different size scales in biology, you're really kind of stuck without understanding, be able to understand biology. So let us talk a little bit about size scale. Okay. So let me get out this. And let's start with a fly. Suppose you had a fly, and how big is a fly? Well, maybe a little fruit fly. It's, a little thing like this, a couple millimeters maybe. So what I want to do is a lot of life that we'll be talking about is at smaller spatial scales. So I want to imagine now, this is a meter stick. Okay, this is a meter stick, imagine. What we're going to do imagine is we're going to try to go down by a factor of 1,000. We're going to imagine that this meter stick is now, so what I want you to do is I want you to close your eyes, and it's kind of corny, but go ahead and go along with me. Close your eyes, and now imagine you're down at a scale where this meter stick is going to be 1 millimeter in length. So now you can open your eyes, and you're living in a world where this is now a millimeter. So that means that, let me get rid of this stuff, that fruit fly is maybe something along like this. Okay, I'm not the best artist in the world. But this is now 1 millimeter. Okay, now at this scale, we're going to be talking about things that are millimeter and down smaller. We don't want to necessarily use fractions a lot, because to me, it's kind of hard to use fractions. So we're going to use another unit. What unit is, if this is a millimeter, then each of these smallest little tick marks that used to be millimeters on a meter stick, how big is each one of those little tick marks?
Micrometer.
A micrometer or micron. So if you look in table 1.1 in the book, there are some metric prefixes that you should know, but the basic idea is 1,000 microns equals 1 millimeter. So this is 1,000 microns. So micrometer is sort of the official term. Micron is the nickname that everybody uses for the micrometer. So let's talk about some other animals and life forms at this spatial scale. You have large single-celled animals. And if you look in the book at table 1.2, it talks about some landmarks for sizes of objects. And sort of large paramecia are on the order of 100 microns in size. That's sort of their basic spatial scale. Now, an amoeba is a little bit bigger than that. It's about 600 microns. So 600 microns is going to be, this is now 1,000 microns, so it's kind of like this. That would be an amoeba. A single-celled animal. So, at this spatial scale, you're beginning to see cells. Now, granted, an amoeba is a pretty big single-celled animal, but it is single cell, so we're down at the level of cellular physics. Not molecular yet, but cellular biology. Okay, you can get even smaller. Paramecium is about 250. So 250 is about like this. Another type of single-celled animal. Here, let me see. I don't trust any of my spelling. You look in the book for that. Other objects are also sort of at this length scale. When you breathe, we're going to be talking a little bit about biomechanics, and in particular, fluid dynamics. Fluids being both liquids and gases. So when you're breathing, that's an example of biological fluid dynamics. You breathe in, breathe into your lungs, and the lungs branch, and they branch over and over like a tree until they finally reach these little end places that are kind of like cul-de-sacs on roads. So here's the branching comes down, and it comes down to about 250. So it's about like this. And it's called an alveolus. That's about 250 microns. And that's where the gas exchange occurs. Another thing we'll be talking about when we're talking in various chapters, including chapter 1 on fluid dynamics and chapter 4 on diffusion, is how you transfer atoms and molecules. And when the gas, this is all gas filled, gets down here, basically moving air in and out of the alveolus is an example of where you have convection. You're blowing, you're breathing in, you're blowing out, you're moving the gas. But then the oxygen actually getting into the blood system is going to be an example of diffusion. And diffusion is going to be the main topic in chapter 4. So we'll be talking about all those, and we'll be talking about the competition of convection and diffusion throughout life. And that brings up the question of how large can cells be? These things are up around several hundred microns. What's the upper limit? What sets the upper limit for a size of a cell? And that gets down to really questions about how physics constrains biology and constrains evolution. And it turns out that one of the limiting factors for the size of a single cell is diffusion. Diffusion works great over short distances, not so good over longer distances. So if you're down here in the order of a few hundred microns, you just can get diffusion to work, and the oxygen can diffuse into the cell adequately. Get it much larger than this, and you can't rely on diffusion to get oxygen inside the cell. So it's really, we're going to actually calculate roughly the maximum size of a cell for a given diffusion constant and a given metabolism for a cell. You can calculate using the ideas of diffusion how big cells can get. And this is about the upper limit before, if you want to have a organism bigger than this, you really got to develop a circulatory system where you can move the oxygen and the waste products around by convection, because diffusion won't be adequate. So hopefully this gives you a little bit of an idea of how we're going to use physics in biology. So we're down on the cellular level. Let's see here. We're going to be talking in chapter 7 a lot about the heart and the cardiac, including electrical properties of the heart and the electrocardiogram. But single cells in the cardiac system are about 100 microns long and about 10 microns wide. They're sort of irregular shaped. This is a cardiac cell. I went to school at Vanderbilt University in Nashville, Tennessee, and we always said that all cardiac cells kind of look like the state of Tennessee. They're kind of long and thin, a little bit irregularly shaped, just like Tennessee. So we'll be talking about cardiac cells and muscle and nerve cells in general. Incidentally, I told you cells are generally hundreds of microns. You always find exceptions, and the exceptions are some of the more fascinating things. For instance, in chapter 6, when we talk about electricity, bioelectricity, the very first, the people who first discovered how cells, how nerve cells work, and the bioelectricity of nerve cells, or Hodgkin and Huxley. And they studied the giant axon of the squid. The squid lives out in the ocean. And this is such a large cell, they were all poke electrodes down into it. So the squid axon is basically this big. That's just part of it. So that's a squid axon. And it's about a millimeter in size, or 1,000 microns. So this is an example where metabolism is not the key thing. It's mainly for signal transmission, but this is a huge cell. That's why it's called a giant axon. It may look real small if you looked at it in an experiment, but if you compare it to the size of other cells, this guy's gigantic. Okay, another cell that's not really a cell at all, but it's more just a bag of hemoglobin, is called the red blood cell. And it's about 8 microns in diameter. So 8 microns, this is down here. But if you look at it edge on, it's sort of a biconcave disc and about 2 microns thick. Now, like I said, these red blood cells aren't really true cells. They don't have a nucleus. They don't divide. They're more bags of hemoglobin that the body produces, and they're what carry the oxygen around through our body. So when we study the circulatory system, we'll be talking a lot about these. In general, cells, this is just a rough number, are about 10 microns in size. I mean, there's exceptions. Cardiac cells even a little large for a cell. Most cells are on the order of 10 microns, so you want to keep that in mind. Now, as you go down inside cells, you can begin to see organelles within cells. Anybody know any of the types of organelles you might find? You've studied biology.
Mitochondria.
Mitochondria is a good example. Mitochondria are about 2 microns in size. So 2 microns would be almost like that. Mitochondria. They are little organelles where a lot of the metabolism goes on in the cell. So you can see these little guys, a bunch of them fit in the cardiac cell, and that's where you have a lot of your metabolism. And so they're about 2 microns. And protoplasts in plants are also about 2 microns. And there's another thing that's about 2 microns, and that is if you go down below cells. What's below cells? Yes.
Molecules?
Okay, what's above molecules but below cells?
Oh, atoms.
Now that would even be below molecules.
My bad.
Okay.
Proteins.
Proteins is getting closer, but they're still largish molecules. You guys got to go up in spatial scale. What makes you sick?
Viruses.
Bacteria and viruses, and in particular, bacteria are the just below sort of cells in size. For instance, E. coli bacteria are about 2 microns in size. They're kind of long and thin. Bacteria are smaller than cells. Most bacteria, if you just had to put a number on them, are on the order of about a micron in size. So it's not too surprising that E. coli bacteria are about the same size as mitochondria, because it's believed that when life first evolved, cells joined with certain bacteria, and the bacteria got taken up by the cell, and then became a symbiotic relationship. But their history of the bacteria, of the mitochondria, go back to independent bacteria that got taken up by cells. So we've gotten down now to a level where we're just a couple of these little tick marks on the meter stick. Things are in the order of a micron in size. Okay. Questions about those things? Well, suppose we're going to go even deeper. So what I'm going to do is I'm going to erase all this, and then we're going to go down one step more in size scale. right, so now you're going to close your eyes, and you're going to imagine that this, which we said was 1 millimeter, now becomes 1 micron. So you open your eyes, and this meter stick's 1 micron in size, which means that E. coli bacteria that we drew before is now this thing. E. coli, and so it's about 2 microns in size. Now, at this spatial scale, we're going to not want to use fractions again, so we're going to use something that's 1,000 times a micron. What's 1,000th of a micron? What's that? Nanometer. So 1,000 nanometers is 1 micron. So we're going to delve down now into the nanoscale, from the micro scale to the nanoscale. Okay. What is smaller than a bacterium, but is still thought to be a living thing? Yes?
Okay, I know I kind of messed up before, but anyway, what about an antibody?
Antibody's close, but an antibody, nobody thinks of an antibody as an independent living organism. It's more of a big molecule. It's going to be on this list, but it's a little smaller. Yeah?
Viruses.
Viruses are on the order of 1/10 of a micron. 1/10 of a micron should be on the order of 100 nanometers in size. And there's one type of virus that was very important in the history of biology and the history of physics applied to biology, and that's a type of bacterium that looks kind of a weird looking thing. It's got a little coat that looks like almost like a stop sign. And it's got little funny legs that come like this, and it's on the order of a couple 100 nanometers. This is called a bacteriophage. Bacteriophage is a virus, but it's a virus that infects bacteria. So some of the very first discoveries about genetics were done using bacteriophages. Because they are really nothing more than a coating with a little bag of DNA in the middle. People guess.
Are all viruses considered living?
That's a good question, because viruses can't reproduce on their own, but they do infect whatever they're infecting and take over the reproductive system of the cell or the bacterium, and then can reproduce themselves. So they're right sort of on the edge of what you consider living. I think there's actually a debate. In my mind, they're a living organism, but you could argue that point all day. Anything more primitive than a virus is basically going to be an inorganic object, and anything more advanced is going to be living, and the viruses are right there on the border. Some other viruses that you may be familiar with is smallpox, which has been eradicated, fortunately. It's a rather large virus. It's about 300 nanometers. I'm very glad the smallpox was eradicated, because it was a very deadly virus. One that hasn't been eradicated but has been in the news more recently is HIV. HIV, which causes AIDS, is about 120 nanometers in size. And you can get even smaller viruses. One that is on the small side, about 30 nanometers. So it's going to be about like this, is polio. Again, nearly eradicated. Not quite. So smallpox, you can say viruses are on the order of 100 nanometers, but you always got to be careful. There's a lot of variation. All right. Now, we can see cells very nicely in the microscope. We can see bacteria, but not very clearly. And viruses, if we can see them at all, are kind of big smudges. And the reason is light. The wavelength of light is on the order of 500 nanometers. It varies depending on the color. But that means light is an electromagnetic wave, and it looks something like this, where this is about 500 nanometers. And you're limited in imaging with light. You're limited for the most part to be able to see things that are larger than a wavelength of light. Otherwise you get diffraction effects. So if you're looking at a cell, which was huge on this scale, remember cells are under 10 microns, which would mean they're essentially filling up this whole wall. You can look at them in quite detail with light. Bacteria, you can see it, but you can't see a lot of detail inside it. And viruses, you really can't see it all, or you just see a smudge, a big blur, the diffraction pattern from the virus. You don't see that it's a virus itself. So we're going to be talking a little bit about, actually, light is going to be covered more in the medical physics class, but it will be one of the issues that comes up when you're talking about physics interacting with biology. Okay. Now let's go down and look at some smaller spatial scales. And let's see what I wrote here. There's other objects that are not living at all. For instance, there's something called a magnetite, or a magnetosome. A magnetosome is about 50 nanometers in size, so it's not too different than the polio. So what it is, it's a little ball of magnetite, magnetic material. There are bacteria that contain these magnetosomes, and usually they contain them in a line, and they're called magnetotactic bacteria. So they get a bunch of these and they line them up, and these cells, these bacteria, E. coli isn't one, but there are types of bacteria that have this magnetite, and they can sense the Earth's magnetic field and move around in response to magnetic fields. That's something we'll talk about a little bit when we get to the chapter on biomagnetism, chapter 8. So we're going to be talking about bioelectricity and biomagnetism. Yes.
What kind of metals are these magnetosomes made of?
It's iron. It's iron oxide, I think. So it's not. It's has to be some sort of paramagnetic material that can sense magnetic fields. So I think it's usually iron. People sometimes claim that you can get some of these in humans, but I think that's still under debate. It's real easy to contaminate samples and get these things in the contaminated samples. So, all right. Now let's go down to some other subcellular organisms. The ribosome. Ribosomes. You know what happens in a ribosome?
Proteins are made.
Proteins are made. So inside a cell, you can have a little object kind of like this size, a ribosome. And really a ribosome, it's about 25 nanometers. Ribosome, you could call it a very small organelle, but it'd probably be more proper to just call it a big molecule. We're getting into larger macromolecules. Ribosomes are made up of protein mainly. That's the protein. Another big molecule of protein is hemoglobin. Hemoglobin is about pretty spherical, about 6. Hemoglobin. About 6 microns. Or excuse me, 6 nanometers in size. So if I ask you on a test, how big is hemoglobin? What's the diameter of hemoglobin? The correct answer would be 6 nanometers. But if you gave me anything 3 times larger, so up to around 18 nanometers or 3 times smaller, 2 to 18, you'd get the full credit, because I really just want you to know that large molecules, these sort of macromolecules, are on the order of 10 nanometers in size. Okay, another thing that you really need to be able to do with biological physics is estimation. So we're going to do an estimation problem, and I'm going to do it over here. You won't be able to see it on the video, but hear it. This is a typical estimation problem would be how many hemoglobin molecules are inside a red blood cell. Because I told you red blood cells are pretty much just bags of hemoglobin. So if I ask you how many hemoglobin molecules are inside a red blood cell, how would you begin to try to answer it? And I just want an estimate. I don't need it to be exact. I want something that's order of magnitude. Anybody want to guess? Yeah.
Just divide the 8 micrometers by the 6 nanometers, but you would just do the hours, not the hours. Just
Be a little more clear on what you said about the powers.
so like it would be 1,000 because you would at least 1,000 because you would divide the orders of power of the smaller, the larger by the smaller.
Okay.
So you wouldn't actually do the math like dividing it, but you would just look at the powers.
You're very close, but you're missing one thing. Yes.
I think we did 8 times 10 to the negative 6 divided by 2 times 2 times 10 to the negative 9.
Okay. You're again close, but not quite there.
Find the volume of each and then divide.
You want the volume, really. It's the volumes, not the length. So this is something we're going to be talking about throughout the semester, about dimensional analysis. If you want to know how many of these hemoglobins fit into a red blood cell, you really want the volume of the two. And that volume goes as the length cubed. So you'd be off by quite a bit just because you didn't raise your answer to the third power. But let's try this. You said the number is going to be the volume of the red blood cell. So how big did we say the red blood cell was?
8 microns.
Well, we said 8 microns. I want to do everything in nanometers just to make our math easier. It's really 8,000 nanometers, but that is, but it's a biconcave disk. So really, I should say 8,000 by 8,000 by 2,000 because it's thinner. So just to be a little more accurate, if you said 8,000 cubed, you'd get order of magnitude correct. I'm going to try a little bit more accurate. 8,000 by 8,000 nanometers by 2,000 nanometers because this thing is this shape if looked from above, but this shape to look from the side. Okay, so that's the volume of the red blood cell. And what's the volume of the hemoglobin?
6 by 6 by 6.
Yeah, you could try to do something tricky like 4/3 pi r cubed, but it's easier because you can't pack these things in too tight. So it's easier to just say 6 by 6 by 6. It's basically 6 nanometers by 6 nanometers by 6 nanometers. And you do then the division. Well, you're going to have 8 times 8, 64 times 2, that's 120. And then there's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 to the 9th nanometers cubed. Down below, it's 6 times 6 is 36 times 6. It comes up to be about 220 nanometers cubed. And the nice thing, the number, the dimensionless number, because nanometers cubed, you could have done this in microns. You'd still cancel out. You get the same thing. Where you can do it in meters. 120 divided by 220, that's about a little over half, about 0.6 times 10 to the 9th. Or, bring this down by the 10 to the 6th, that'd be 600 million. So you could, if there's nothing else in the red blood cell besides hemoglobin, it's packed about as tightly as you could fit it, you'd have on the order of 600 million hemoglobin molecules.
So we can assume everything has like a cube shape instead of like a...
If for an order of magnitude estimate, that's a safe assumption. Now you always should be thinking when you make these estimates that these are approximations and it's not going to be exactly right. But if you want to know whether it's 1,000 versus a million versus a billion versus a trillion, that's pretty good. That'll get you a good estimation. So whenever in the homework you're asked to estimate something, I'm asking you basically to make some assumptions and get a rough guide to how big something is. If I'm asking you to solve a particular problem, I might be wanting something a little more accurate. But when you see that word estimate, that means you want to do it rough and dirty, but you want to get an order of magnitude right. And so this tells you something important. This tells you basically you can have You can have about, I think, 4 oxygen molecules bound to each hemoglobin molecule. So you can basically calculate the concentration of oxygen in the blood knowing this number. So it's a very useful thing to have, to be able to do these sorts of estimates. Questions on the estimation problems? First couple problems in your homework, you'll be doing some estimates, similar types of estimates. Okay, let's continue our look at different spatial scales. There's another, I've been drawing like the borders of all these objects. It's just a thin line, but in fact, cell membranes are what separate the insides and outsides from cells. And cell membranes are about 7 nanometers thick. So cell membranes on this spatial scale look sort of like this big. And when we talk about bioelectricity, we'll be talking a lot about cell membranes. This is what kind of molecules makes up the cell membrane? Anybody know?
Phospholipids.
Lipids or fat. So it's a bilipid layer. It makes up a cell membrane. So really, this outer coating of the E. coli is this thick. But there's often proteins lumped into the membrane as the proteins do all the interesting things. Yes?
It's not a 7, it's a little hard to tell.
It's a 7. Yeah, 7. Some of this is a little clearer in the picture in the book, which has a lot of these in it, but that's a 7 nanometers. If you go down even smaller and you get down around 1 nanometer, you're getting down to the size of reasonably small molecules, not tiny, reasonably small. For instance, glucose is about 1 nanometer. Remember, 1 means you're going to be down at that smallest tick mark on the stick now. Glucose is the main metabolic molecule that we get our energy from. So it's got 6 carbons, 6 oxygens, 12 hydrogens, something like that in it. So we're almost down to the atomic scale, but not quite. ATP is another molecule that's roughly this size. It's important in metabolism. So you want to go down to the 1 nanometer scale, you're down to molecular sizes. We've gone from cellular down to molecular. Suppose you want to go down even smaller. What's the most common molecule in all of life? No, not carbon. Carbon, first of all, that would be more of an element, yes?
Water.
Water is what we're all made out of. Water is H2O, so it's just 3 atoms. The bond length between the oxygen and the hydrogen is about 1/10 of a nanometer. So I basically can't even draw it. Water would look like this. There'd be a bunch of water molecules on this spatial scale. So if you lived on the 1,000 nanometer scale, so this is 1 micron, you would barely be able to see atoms. It would look very grainy, but you would be able to tell that there's some graininess in the world. Individual molecules like water, you'd be able to tell that it wasn't a smooth continuum like it looks like in our spatial scale. If you wanted, sometimes people use a nickname. Water is basically a 1/10 of a nanometer, and there's a special name for 1/10 of a nanometer. You guys know what 1/10 of a nanometer is?
Armstrong.
Angstrom, yes.
Angstrom.
So 1/10 of a nanometer is 1 angstrom, and a lot of the old literature will specify sizes in angstroms. Angstroms are going out of style. There is this committee that's sort of the unit police, and the unit police want units that go down in factors of 1,000, milli, micro, nano. So if you're using angstroms, it's frowned on these days. But you'll still see it a lot, in especially the older literature. Angstroms are basically the size of atoms, because water is just 3 atoms. So atoms are 1/10 of a nanometer in size. We can keep playing this game, but you really have to go a long ways in order to see something even smaller than the atom. Because we will talk in medical physics class next semester about nuclear physics. Nuclear physics goes down 10 to the minus 15 meters is the size of a nucleus of an atom, cell of an atom. 10 to the minus 15, that would mean 10 to the minus 6 of nanometers, which means that that's 1,000,000 times larger than a nucleus of an atom. So nuclei are really, really small. But we won't get to that this semester. You have to study it next semester. Now, if you like this sort of thing, I would suggest a couple books. Okay, I'm going to be suggesting books to you all semester. Okay, I'd love for you to take it some days to read them. I realize you don't have time to go out and read all the books I suggest, but I just want you to know the types of books I think would be useful to look at. One is called The Machinery of Life. I have the first edition. There's an updated edition that's even more colorful. It's by a man named David Goodsell. All these books, I'll send you the links through Moodle so you can find them. It shows a bunch of pictures of different molecules, and they're all drawn to scale. So they're basically says like this is, here's hemoglobin at a 10 million times scale. So you can actually measure this with your meter stick, and then if you divide by 10 million, that's the size the hemoglobin molecule is. And there's a little version of it that's 1,000,000 times the scale. So it gives you a really good sense for the relative size of things like we've been talking about. It's also a great introduction to biology. So if you know no biology and you wanted to learn a little bit of biochemistry for the class, you might want to just take it. It's well written and it's easy and it's short. And you can get Machinery of Life. And I think the library has a copy. But it's one good book. Another good book, older one, is called Powers of 10. And Powers of 10 was written by Philip Morrison and some of his collaborators. He was one of the men who worked on the atomic bomb and the Manhattan Project. And what it does, it's a very famous book. It has each page has a different picture. It starts in Chicago, a park where a couple are sitting and having a picnic. And if you turn the page, that's a 10-meter scale. You turn the page, and now you have the 1-meter scale. And here's the couple at the park, and then you go down, and now you're looking at just the hand. Each page is a factor of 10. So that's why it's Powers of 10. You go down, it also goes in the upward direction, which we didn't talk too much about. There they are at the 100-meter scale. I don't know, anybody here from Chicago? You might recognize things now. This is, that's the stadium where the Bears play, Soldier Stadium, I think. I believe that's the planetarium. You're getting into Chicago, that's the kilometer scale. So if you've, here is 10-kilometer scale. Now you see Lake Michigan. If you're a 5K runner, halfway across that is the length of your race. And you go on up, and it goes all the way up to, the galaxies and things. So if you like this kind of thing, this is a great book. There's a video online that goes with it, so I will send you that link to the video too. So it's just a couple of ways you can get a better feel for the sizes of things, both biological and non-biological. All right, so that is today's class. It's really talking about that very first section. We will start talking about biomechanics on Friday. So if you can, try to read sections 1.3 to 1.8. Any last chance for questions? Anything you thought of that you're sure about? Yes?
So you've been working in this field for a while now. These measurements and numbers that have come out since you started.
Well, millions, but that's just because people keep measuring new and new objects that are slightly different from other ones. I don't think any real fundamental new spatial scales have been discovered, but people who study bacteria all of a sudden find there's a million different species of it, and each one's got its own size. So it really depends on how you count. But yeah, it's a fascinating subject. Anything else? Other questions about the syllabus and how they do it? Okay, in that case, I will see you on Friday. Great.